All right. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. Next slide, please. This webinar is brought to you today by Plantae, the open online community for plant science powered by the American Society of Plant Biologists. I'd like to give a special thank you to all our ASPB members who are attending today's webinar. Your ASPB membership dues help support making this webinar possible. For any of you who have not yet joined ASPB, you can join today using the discount code %10 to receive a 10% off discount on your membership due. You can learn more about ASPB and the opportunities that we provide at aspb.org. Next slide, please. This webinar is recorded, and we have so many other webinars that we've done over the years. So if you're looking for plant science content and are looking for great resources, be sure to check out our Plantae YouTube channel, and you can find all our previously recorded talks there. Next slide, please. Before we get started officially, I'd like to go over a few housekeeping items to make sure that you get the most of your attendance today's webinar. If you are experiencing technical issues, let us know about those using the Q&A box, or you can email me at jpadilla@aspb.org. If you have questions for our guest speakers, please add them in the Q&A box of your Zoom sort of control panel. Our moderator will be sharing them with our presenters. If you have trouble or need to leave the webinar early, as I mentioned, this webinar is recorded and you'll be able to watch them on our YouTube channel once it's processed. So be sure to follow the channel to receive notification when the new content is posted. Without further ado, I am now going to turn it over to our moderator who will facilitate today's session. Take it away, Plantae Fellows.
Thank you, Jason, for the introduction. Hello, everyone. I'm Rini Rahman, and I'll be moderating today's webinar along with two other Plantae Fellows. We are delighted to have you all with us for today's session on how to build a productive lab. As early career researchers, we often focus primarily on the scientific side of lab work, which includes designing experiments, troubleshooting protocols, collecting data, and analyzing them. But behind every productive research, there's a strong operational structure in terms of effective people management, effective budgeting, administrative coordination, and the ability of a person to oversee multiple projects simultaneously. These elements, although less visible, play a crucial role in shaping the lab culture and productivity. To help us understand how these components come together in a real lab environment, we are joined today by two accomplished research professors who will share their experience in setting up and running their labs. They'll offer insights into decision-making as a PI, managing diverse student projects, maintaining consistent publishing pipelines, navigating conflicts, and strategies for forming networks within the science community, all of which contributes significantly to the construction of a productive lab in the long run. Next slide, please. So before we begin, let me briefly walk you through today's webinar structure. We will start with moderator and speaker introductions, which will be followed by the speaker presentations. After both the talks, we will continue to a Q&A session. The audience, please feel free to submit your questions in the Q&A box at any point during the seminar. If you prefer, you can also address questions directly to our speakers during the Q&A session. And last but not the least, we will end with a short concluding remark. Next slide, please. All right, so today's moderators are myself, Rini Rahman. I am an ex-postdoctoral researcher from the National University of Singapore. I just moved out of Singapore in November this year. The other two moderators we have are Mohammad Aslam, who is currently working as a research scientist at Donald Danforth Plant Science Center, Missouri, and Abdul Kabir Omiza Abdul Malik, who is a PhD student at HHU, Germany. Next slide, please. Now I would like to invite Mohammad Aslam to introduce our speakers for today.
Thank you, Rini. Hello, everyone. I am Mohammad Aslam, and I'm delighted to serve you as moderator today. It is my great pleasure to introduce our first speaker, Dr. Carl N. Dr. N is a professor of plant biology at University College Dublin, where he leads a research program focused on plant cell signaling, particularly in the context of abiotic stress. His lab has made significant contributions to our understanding of sphingolipid-based signaling, guard cell and stomatal biology, and how plants perceive and respond to challenging environmental conditions. Beyond his academic research, Dr. N plays an important role in the scientific community as an editor at New Phytologist. He is also deeply committed to mentoring and teaching, coordinating core modules such as Introduction to Scientific Research and Cell Signaling in Plants at University College Dublin. Today, Dr. N will share his insights on building a lab environment that is not only scientifically productive, but also resilient and supportive, drawing on his experience both as a researcher and as a steward of scientific publication. Please join me giving a warm welcome to Dr. Carl N. Dr. N, thank you for being here. Over to you.
Good day, everyone. Thank you, Rini Mohammed, for the kind introductions. I'm delighted to be here. I'd like to thank the Plantae Fellows for inviting me to give the seminar today, to share some of my experiences, and a big shout out to Jason for all the technical aspects of this webinar, which I hope will run very smoothly. So I've been asked to share some of my experiences on efficient people management and publishing, and I hope I'll be able to share with you some of my own experiences that will hopefully be able to help you along your scientific career. So just a little bit about myself. So I consider myself as a global scientist. I've gone from east to west. So on this slide, my training is in botany at the National University of Singapore. I'm a Singaporean by birth. I'm still a Singaporean, a proud one, I guess. So I did my Bachelor of Science and Master of Science degrees at the National University of Singapore. And then I did PhD on stomatal biology at Lancaster University in the UK. And that followed by a postdoc in Penn State. And then I answered Ireland's call, and I moved to Ireland, a beautiful country, very green and very wet as well. And I'm currently a professor of plant biology in Dublin, in the Republic of Ireland. So I guess before we even start about people management, I think it's important to hire the right team. The right team really makes a big difference for the lab. And the approaches I would use in relation to hiring the right team would be to advertise the position as widely as possible. So by doing that, you really want to capture the global scientific talent. And from there, hopefully, you'll be able to then find the right person for your team. Now, of course, when you advertise as widely as possible, it's also important to specify very clearly what are the requirements for the position. So this will vary whether you're looking for a PhD student or whether you're looking for a postdoc or a research fellow. So be very specific in what you're looking for. That really helps the candidates that are applying. It also helps you to manage the number of applicants as well. Now, of course, when you hire people for your team, it's really important to be aware of unconscious bias. So, this can take the form of gender bias, ethnic bias, and affinity bias. When we all suffer from that. But I think it's really important to familiarize yourself with unconscious bias and how to remove that during the hire process. Now, depending on the position that you're hiring, whether it's a PhD position or a postdoc position, you really have to then assemble a panel to help you in the shortlisting and interviews. I do not recommend that you do the shortlisting on your own. It's really important to have different perspectives, and also not to conduct the interviews on your own. Make sure it's a panel-based interview. Of course, when you take into account the composition of the panel for the interviews, then you also have to consider EDI, or over in the States it's DEI. I know that's not quite a popular term at the moment, but it's really important to take into account equality, be diverse, be inclusive. So for a PhD position, what I typically would do is, as a supervisor, I would be on a panel. I would invite co-supervisors if there are co-supervisors on the projects, a postdoc from the group if available, and a senior academic colleague to be part of the shortlisting and interview panel. And now for a postdoc, it's slightly different. I would invite co-mentors and a more senior academic colleague as well. Now, I think it's important to actually conduct the interviews in person, if possible. I know sometimes it's really difficult if you're from the other side of the world. But if not, then you can do it remotely. Not the best, I think. Remote interviews, in-person interviews are probably the best. But definitely obtain references. This can be in the form of written references. I think verbal references are actually very, very good. So you could do both. But the key thing is, do not be hasty in hiring decisions. So, for example, you have shortlisted the candidates. If you interviewed them, if they're not suitable, do not be desperate enough to hire. Go for another round of shortlisting and another round of interviews. You really want to make sure you get the right team in place for your project. Now, once you have the right people for the project, now onboarding is really important. So this is probably the most difficult part of the experience for the new team member. They may not have come from the same department. They may have moved countries to join your lab. So it's very important to set clear expectations, at least for the initial period. Initial period can last between several weeks to maybe 2 or 3 months. Have a good discussion with them about what they should focus on. Especially for PhD students, you really need to assign the readings to get them started with the research. Of course, when you're onboarding a new member, you have to make sure the necessary resources are available. It's also important for them to familiarize themselves with the key personnel in the department. So who's involved in administration, who to go to for technical support, etc. But really important thing is to check in with them regularly. Don't just leave them on their own in the lab. And ensure a supportive environment. I think a good way to do this would be to assign a lab buddy, if there's one available. So a lab buddy is really useful because it can assist a new member in terms of lab logistics, like use of equipment, spaces. They will be familiar with what the university resources are, so they can actually help the person to settle in the initial period of the position. Now, of course, you also have to then be aware of cultural differences. Different cultures will approach problems quite differently. They approach interactions with people quite differently as well. So it's really important to do that, and to encourage networking, not just within the group but also outside of the group. PhD or postdoc is where you really start to build a network, and it's really important to help them along with that. And I think manage expectations. So do advise your lab members to take breaks or holidays. Please do not force them to work 24/7. Okay, so I think it's also important now that you have a team in place to really recognize your roles and responsibilities. So my approach is the PhD students and postdocs are unique individuals. So they are unique. So not two persons are the same. So it's really important to take time to recognize their strengths and their weaknesses, because once you know their strengths and their weaknesses, you're then able to manage problems that may arise. Some of your students may be great at very, very technical experiments, but maybe not so good with overall big picture view of the project. So, once you're familiar with that, you can address them. Really make time to meet with them on a regular basis, and they sign up to do a project with you. So don't leave them in the lurch. Make sure you meet with them. I see my students once a week at the very least. Or what I tend to do, I like to go down to the lab and just nose around and have a chat with them about what they're doing. A postdoc then would be somewhere in between a PhD and a PI. I see a postdoc as maybe a transition period between you finish the PhD, they're doing now a postdoc, and hopefully they will then transition to a PI at some stage. So they're a good interface between the PI and the student, and I cannot stress enough in the importance of work-life balance here. And really do not treat your PhD students and postdocs as another pair of hands. They are really, key personnel of the lab that you should really try to mentor quite well. Just remember, they are going to be the next generation of PIs that is going to make all these exciting scientific advances. Now, I've been given a few topics to cover as part of the webinar. Strategies on handling multiple lab projects simultaneously. I think that the key message, key take-home message for this is really time allocation. So time allocation on the project will depend on the personnel associated with the tasks of the project. This could be PhD students, postdocs. The PhD students will require you to devote a bit more time than a postdoc. Postdocs tend to be more independent because they would have had the experience of doing the PhD. And the time allocation then should really be based on the project milestones and deliverables. So, you should really be very clear with the team members on what are the milestones and what are the deliverables and the timeline for those. And once you have that quite clear, then you can really then, be quite good at then managing different projects in the lab. And in terms of assisting career development of postdocs, I think this also covers PhDs as well. In manuscript writing and project development, I think I've never had the experience of attending a writing workshop, but I think that could be quite useful. But I think the best way to learn to write is really by reading the published papers. You do learn a lot from the style of writing and the messaging that has to go into writing papers. In terms of career development, core peer reviewing would be very good for postdocs. PhD students as well, if you think they're up to the job of peer reviewing. I was very fortunate. I had the opportunity to co-peer review when I was a PhD student, and I really enjoyed the experience. And I strongly encourage, if you're a PhD student or postdoc, do definitely go and discuss with the PI about co-peer reviewing of papers with them. I think it's very important if they're going to be the next generation of PIs that you maybe share your successful grant proposals with them, so they know what's involved in structuring a successful grant proposal. And in terms of manuscripts, be patient. I think be prepared to go through multiple drafts or multiple rounds of editing before the final submission. The last thing you want to do really is to submit in haste. And the reason for that is you need to consider time devoted by the reviewers and also time devoted by an editor. So, we are all humans as well. So, you try to make the process a bit more straightforward for everyone. So do not submit in haste. In terms of publishing strategy, this will vary from group to group. It will vary between PIs. I think definitely go for open access. Open access publishing is really important. It really gets the science across to a global audience. So it ensures wide readership. Of course, in terms of publishing, affordability is a big issue here. I bring up example here, the APC for Nature. I mean, looking at APC for Nature, at least from an Irish perspective, that's nearly half the stipend of a yearly PhD. So, you need to consider whether the group can afford the APC or whether the funds can be channeled to more useful experiments that will also result in a very, very good paper, but may not be, may not be published from the Irish perspective and for European perspective. DORA is a big one for us. That's the San Francisco Declaration on Research Assessment. So this really removes the journal impact factors in terms of for measuring quality of research articles. So what we don't do anymore is to use journal impact factors to assess individual contributions. That we apply that in hiring, promotions, and funding decisions. So we don't use journal impact factors anymore. I think in terms of publishing, I think for me personally, I like to support society-based journals and not-for-profit organizations. And the reason for that is they really give back to the community. I think it's very important to do that. So I list here two of my favorite organizations here. I know ASPB. I have to mention ASPB. I think ASPB does a great job, and they publish really good journals like the Plant Cell and Plant Physiology, and they really give back to the community as well. I mean, Plantae, for example, the webinar is supported by ASPB. Also, I work a lot with the New Phytologist Foundation because I'm an editor for New Phytologist. They also publish, very, very good journals like New Phytologist and Plants People Planet. And they also fund a lot of symposia and workshops, travel awards, and one of my favorite ones that they do is the Next Generation Scientist Symposium that they run yearly. It's really to encourage the young career scientists to really come together to network and share their. I think in terms of publishing strategy, a lot depends on the budget that you have in the lab, but do consider supporting the not-for-profit or society-based journals. And in terms of writing grant proposals and securing funding, that's a really challenging one for everyone. I think whether you're early career PI or whether you're intending to apply for a fellowship or you're more senior academic. But I think there are several principles or key take-home messages here. Read the funding program call document in detail. Okay, do not skip sections of the funding call documents. Now, depending on the funding agency, the funding call can be either investigator-led. This is really a bottoms-up approach where the investigator decides on their favorite topic that they want to apply for funding for, or it could be a policy-driven approach by the funding agency. So it's a top-down approach. Then they might specify particular research topics or particular areas. So you really need to understand, and you get that information from reading the funding call document. An amount of time that you spend preparing the proposal will also depend on whether it's a single PI-led program. So for example, I'm a PI, it's a single PI-led program. So I'll do all the writing on my own, as opposed to a consortium program where you have multiple partners, and sometimes there are multiple partners from different countries. And that's where it takes a bit more organization to get everyone to contribute their particular sections to the proposal. Really start preparing early. Do not leave it to the last minute. I think as a PI, if at all possible, you should try to submit at least one research proposal a year. Funding success rates are quite low. So, you really need to submit one to kind of give yourself a chance to get some funding. I thought it's important for me to also talk a bit about the narrative CV. I know that's not quite over in the States yet, but if you're based in Europe or you're based in the UK and Ireland, we do not do traditional CVs anymore for grant proposals or fellowship applications. It's what we call the narrative CV. So it's a way to move beyond the traditional list of publications and metrics. So what you're not allowed to do in narrative CV is to specify the journal impact factor. That's a big no-no for us. And And in the narrative CV, then you use written descriptions to highlight your contributions and achievements. So you really need to tell a story about how impactful your research is. I've listed here some of the funding agencies that I'm familiar with. I know none of the American ones are listed here because I'm not based in America, so I'm not familiar with at all with the North American funding system. But a lot of the so over on the left here, these are the agencies that will adopt a PI-led, a bottoms-up approach. And the key ones would be the European Research Council, the Marie Curie Fellowship, for example, the Royal Society, BBSRC, and Research Ireland. Some of these also do a top-down approach, so where it's a policy-driven sort of research where they do specify the topics of the research call. So typical would be big funding agency for the European nations. And again, in Ireland, the Department of Agriculture, Research Ireland, and in the UK, the BBSRC. I think that's, and I have one more slide. Hopefully, I'm not taking too much time. That relates to manuscript authorship. This is an important one because papers are our currency in academia. In relation to authorship, that can be quite tricky sometimes. So there are a few no-nos. So authorship should not be based on seniority or position. So you only include authors where they have contributed. You do not include authors on the basis that you think they can improve your chances of paper acceptance. Don't include your friends just because you want to help them to progress their career. I think in terms of assigning authorship, the lead PI should take overall responsibility. There's really I don't think there is community consensus on how to do this, but I think it's really important to be open and consistent. So adopt a consistent approach to this. Scientific integrity is really important here, especially if you're a new PI or you're early career PI. It could be useful to take advice from senior colleagues on this aspect of assigning authorship. And of course, does author list position matter? I think in the plant sciences, it's generally accepted that the first and the last authors are the most coveted positions. But of course, not to forget that there's the possibility of joint first authorships and also joint corresponding authors as well. Now, from time to time, you may get a dispute in terms of authorship of publications. I think this should be dealt with early on, to not leave it to fester. I think if there's a dispute in terms of authorship, discuss internally, first among the senior authors, and then with all authors involved. But it's important to be transparent. Assign authorship based on relative contributions. I want to highlight a very, very excellent insights from Sophia and Kamoon here. I provided the link. I'll definitely go and have a read on that post where Sophia actually discussed authorships and providing very, very useful advice. And I think that's the end of what I'm supposed to be discussing today, and I'm happy to take questions later on after Yuchen's talk. So thank you very much.
Thank you, Dr. N, for an excellent presentation. Before we move on, a quick reminder that you may submit your questions in the Q&A box at any time. Now it's my pleasure to introduce our next speaker, Dr. Yuchen Long, Assistant Professor at the National University of Singapore. Dr. Long completed his PhD in plant developmental biology at Utrecht University in the Netherlands. His postdoctoral work focused on the biomechanical regulation of Arabidopsis shoot development. At National University of Singapore, Dr. Long leads a research program at the interface of biology and physics, integrating molecular biology, live imaging, micromechanical measurements, and theoretical approaches to understand how mechanical forces shape plant development. His team investigates how turgor pressure, tissue hydraulics, and mechanical stress interact with genetic and signaling networks during morphogenesis. Dr. Long's work is especially relevant to our webinar theme because building a productive laboratory often depends on creating systems and infrastructure both physical and organizational, that supports interdisciplinary research. His insights into mechanobiology, quantitative experimentation, and lab design will be valuable for anyone considering how to structure a modern integrative research group. Please join me welcoming Dr. Yuchen Long. Dr. Long, thank you so much for being with us. Over to you.
Yeah, thank you very much, Mohammed, for the introduction, and thanks a lot for the invitation by the Plantae Fellows. So let me start my share screen. So I'm happy today to actually share a bit from my experience about boosting lab productivity through networking, collaboration, and publicity. A short introduction of myself. So I was born and raised in Beijing in China. And after high school, I traveled to the Netherlands to complete my Bachelor of Science as well as PhD training in both the Han University and the Utrecht University. The lab later on moved to Wageningen, but actually my degree was given by Utrecht. So yeah, this is where I put down here. Afterwards, I moved to France to do my postdoc training in the ENS de Lyon, and I think I took a rather brave step for myself to move from pure biology to the interface between biology and physics, and actually joining a lab which is headed by a Professor Areski Budaud, who is actually a physicist by training. And in this lab, I think I really learned a lot about how to work together with a group of interdisciplinary researchers together under the same roof, as well as across continents. And with all the experiences and all the encouragements that I got throughout the years, I finally decided to actually return back to Asia, to Singapore, at the end of 2020. And if you still remember, that was actually by the middle or towards the end of the COVID-19 pandemic period. So actually, the move was quite tough, but I need to give a lot of thanks to both my previous host, University ENS de Lyon, as well as my current host, the National University of Singapore. They really helped a lot in helping me to smoothly transit between this process and establish my new lab amid this very challenging time. Yeah. And having a rather diverse background and also trying to build a team that have different opinions and different voices, I think we have been trying to also build a multicultural, multinationality lab. And so far, we have already been able to host, I think, a researcher from 9 different countries, as listed over here. And we believe that we are also going to continuously boost our diversity in the lab in the future. One thing I think it's counting ourselves lucky because right now I think biomechanical or even maybe a very specific interdisciplinary studies as we do in our lab is not that commonly found around the world. So for actually candidates who really wanted to work on very specific topics, we are one of the few choices out there in the world. So I think that's why we've been so lucky in being able to welcome so many different people from different backgrounds. And of course, at the same time, I want to acknowledge that, the nationality and the training background is not the only part of inclusiveness. And I think there are a lot of things that we still need to work on to actually improve, how everyone can actually share their voices freely in the lab. Because I think the most important thing is that everybody can actually enjoy what they do and also actually have a moment to shine in a team. So as Mohammed introduced us, so our lab is actually at the interface between biomechanics and biology, and actually that's why our lab is called the Mecha Plant Lab. And a small trivia: this name is actually voted by the lab members. So don't ask me why or how, but anyways, our lab is interested in 2 distinct, very basic questions. That is, how large can a plant grow to, and also what shape can plants grow to? And we actually focus quite a lot on how plant cells generate mechanical forces and also perceive forces from the outside, thereby actually shaping itself as if they're sculpting using their cells. Our initial research priority was given to the fundamental research of development, and we focused on Arabidopsis thaliana since the beginning of our lab, even during the pandemic before even the first lab member arrived. But over time, we realized that actually there can be a lot of interesting revenues out there. And more importantly, when we were welcoming new lab members, actually some of the postdocs had very strong opinions of working on some completely new models that I have never worked with. For example, one postdoc came and convinced me that actually a liverwort or Mercantia polymorpha is a great model to study mechanical and about developmental regulations. And another lab member says, but actually I think Fiscomitrium patens moss is the best model. And as the opportunity provided itself, I thought, why not we just pick them up on the way and then actually see what we can do. And I think even though the current work is still ongoing and we don't have anything published in these new organisms yet, I believe actually this is a great way for us to not only move beyond what we were able to do, but also actually like stimulate the motivations from the lab members, because these are exactly what they wanted to do. This is what they came to do, and actually by actually building on their motivation and enthusiasm, we're now I think quite comfortable and confident in implementing more models. So very recently, through an internal collaboration with another labs at the National University of Singapore, we also started a small project that is working on duckweed or Wolfia globosa. Oh, sorry about the typo over here, globosa. I think it's maybe an auto correction issue. But anyways, I think because we were able to establish these new models early on in our lab, we're now very confident that we can do something about that. And hopefully, in very near future, we can share some new findings on these projects that are all actually spearheaded by new lab members who came with very original ideas. Yeah. So after moving to a institute, especially during the COVID pandemic, it was quite difficult to establish new connections. And I think in our university we had quite a few plant labs, although at the same time, the labs were working on very different stuff. And then at the beginning, I actually missed out this process of integrating into the institute. So try to cope with this and also help myself in getting to know each other. Me and a few other PIs, including Ong Sang Lao, actually the previous supervisor of Rini here, as well as Dr. Enyang Che, who recently moved to Oxford University. So we came up with this idea called the DBS, the Department of Biological Sciences Plant Forum. So this is a monthly meeting that actually didn't exist before, of course. And then this meeting is an internal seminar series that is, organized by the junior researchers and for the junior researchers. So very often we will recruit maybe one postdoc and one PhD student to work in a group to invite internal and external speakers. Most of them are actually also on the same level of their seniority in research, that is basically students and postdocs, to share their ongoing work, their even work plans, or maybe previous work to actually build and establish an environment of exchanges. And of course, we, if opportunity provides themselves, we also invite external speakers. And this month, actually, we have the luck to welcome Dr. Stéphane Verger from Umeå University, who is actually visiting our lab for 1 month right now. So this is within our department and actually on the university campus with a different institute, the Mechanobiology Institute. We also established a interdisciplinary meeting group that is called the Tissue Mechanobiology Monthly Meeting, or for short TMM. So I think the stomato people in the audience will maybe get the wink wink nod notch over here. But anyways, so the idea here is that even though these group of scientists, they're actually coming from very different backgrounds. Some of them are working on animal tissues, some of them are working on disease models, some of them are real physicists, and actually we are the only plant lab in this community. But because we all have a shared interest in mechanobiology on the tisular level, that's why we decided to actually group together and then share our different perspectives and our ongoing work on a monthly level and on a monthly basis. Yeah, and I think at this moment, this meeting, both of the meeting, I think has has been going on for 3 or 4 years already. So, and it's actually getting quite a thriving and helping our lab members to gain confidence and also actually practice for presentations before they maybe go on to conferences or more importantly, go on to interviews for their next position. Yeah. And during these meetings, we also actually meet new friends. So I want to actually highlight one of our very recent collaborations with the Mechanobiology Institute lab headed by Jennifer Yang. One of Jennifer's PhD student, Avery Sun, who was actually working on animal-based biomaterials, came to us because after one of our joint TMM meetings, we actually demonstrated a new technology that we have implemented in the lab, that is to measure, basically doing a confocal microscopy and atomic force microscopy in a simultaneous way. And they said they believe that actually can be very helpful for their study. So then actually we established a small collaboration team within our lab and Jennifer's lab. And together, this work has contributed to a very great piece of work that is recently published in Nature Materials, headed by Jennifer here. So I think the publication indeed is great, but what I wanted to really highlight is actually the clashing of different ideas. Someone that is coming from material science and actually animal biology with someone that is purely working on imaging and plants. But then actually by putting the ideas together, we found something very, very interesting. And it's also a great demonstration of the new technology that we are building. So quite interestingly, you know, as a plant lab, this is actually our first research paper coming from from us, which is not on plant at all. But I think it's not a bad thing. In fact, it's also shows actually how versatile I think and how promising interdisciplinary researchers could be, provided that it's actually built on a collaborative and interactive culture. Yeah. And maybe going a bit further on how to establish the research network, I think I only speak from my own experiences. I think the easiest thing to do is to actually cherish the environment that you have been in and you are going to. So one thing I think is very important is to maintain your connections, your previous connections from PhD and postdocs, or maybe even earlier educations, to actually maintain this great network where people know you, they know your quality, and also actually you know their work, and therefore maybe ideas can simultaneously emerge. But of course, at the same time, I think in some cultures, it is kind of frowned upon a little bit, but to actually immediately continue to collaborate with their previous supervisors because they might be seen as a lack of independence. So I think depending on in which institute, in which country you are, you might want to keep that in mind, how you want to approach this kind of networking in the future. The next thing is to definitely reach out to the new colleagues at your host institute, and I think in our case, National University of Singapore is a very it's a big university with a lot of different researches going on. So it was actually quite easy to already entertain ourselves inside the campus, and also maybe reaching out to our colleagues at the Nanyang Technological University in the same city in Singapore. But I think besides these two, which are very natural. The next simplest thing to do is to just go out and approach people in conferences. Since you're already there, and since there are so many like-minded people who join the same conference, why not just talk to them? And of course, here I highlight a few different conferences, which are not the only ones that I've been to, of course, but I think they meant a lot to me on different levels. The SFBD and the ECR 2021, I think, were both virtual meetings amidst the COVID-19 pandemics. And I think really thanks to meetings like that, it was able to stay in contact with the community and actually get inspired and also get encouraged in the works that we do. And then I think it was 2023, if I'm not wrong, I think the New Phytologist Next Generation Scientist Conference was held in Singapore. And actually, this is the first time that I met Carl in person, as well as also the first time that I worked closely with Rini for conference organizations. And I think kind of helped me to get the opportunity to also share this experience with you here. And the other meetings, for example, this is the first national conference of plant stem cell biology in China that actually helped me to connect back to my roots with my origin, as well as the IPMB, as well as the new webinar that is leading to the next biomechanical conference, which is the largest one, I think, for plants that is going to happen next year in the neighboring country of Indonesia. Yeah. And I think particularly in this the new phytologist, next generation scientists, where by the way, I see Carl over here and I see Rini here. Yeah, I think it was first of all one of the first meetings I had in person after the COVID pandemic, and was actually very interesting for me because I was more involved in this meeting because one of the invited speakers couldn't actually join because of personal reasons, and since I was among the local team, so I was actually called in to fill in the spot. But then I thought, okay, well, I'm gonna take this opportunity and I'm gonna really deliver what we can and also make all the connections that we can. And indeed, I think in this case we kind of convinced, editors from other journals, and very soon later we got invited to actually write a small piece of textbook style review or primer article about physical concepts in biology, which we think it's actually very helpful for the community, we believe, but also very helpful for our lab to actually establish an image of us before actually we even publish our first research paper. So I would say, definitely make the chance to go out, meet people, and make connections, and actually you never know what will happen to you. And I think most of the time it's actually a win. Finally, I think I want to touch on the establishment of the lab website. So actually, we didn't start with the functional site until when we started the first round of lab member hiring. And I thought, okay, maybe this is the moment that people actually should know about what we're doing, but also at the same time, actually, the rest of the world should know actually who are in the lab. So we actually used a WordPress site for our lab. And I think there are generally different strategies. Some universities actually have their dedicated personal pages or lab-based pages where you can customize, or maybe the university team can help you to generate. So in our case, I think we decided to make our own site so we can be much more autonomous. But at the same time, if you're doing this, do remember to include any disclaimers that because this is not an official website, that everything that you claim is actually representing your own opinion, but not the institutes or the universities. And I think this can help you to save a lot of the issues. And the reason I also say that is that if we look at the statistics, actually most of the people who landed on our website actually is through the National University of Singapore website. So indeed, they would actually continuously, with the continuous flow maybe of thinking that this is actually part of the official web, but then actually this is something to our lab alone. I can also see here where the other sources of the clicks that go into the website. But I think more importantly is that actually why people come here. What did they actually view? And quite interestingly, you can see most people actually came here to check who are in the lab. It's on the people's page. So I think I just want to quickly show you how it looks like. So if we come to this website, we have some news posts over here. But actually this, I think, is indeed the most important page. This is where every single person have their photo, if we have the photo, and also their names over here. And the reason is because quite often, you have lab members to go to conferences where maybe people remember their names but do not remember how they look like, or maybe they remember how they look like but don't know actually who to contact. So in this case, it is actually a great way to expose your lab members to the rest of the world and actually for themselves to be recognized by the works that they have done. And I think here everything else, our research, our news, all these things can show up on social media, but the only thing that is very rarely seen is actually your lab composition. And I think in our particular case, previously our department did not have this page for our lab alone, and that's why we decided actually to include. So actually, this whole website is just an excuse to make this lab member list. But I think as it shows, it actually pays off. And then actually during this process, we also even hired more people who actually visited our website and then also attended conferences, meeting us, etc., etc. Yeah. So I think with that said, I just want to close off my short talk over here, and I want to give my acknowledgments to the current lab members: Kartik Babu, Xin, Constance, Noemi, Xin-Yi, Wacharapon, Pakwan, Chao Wei, Malcolm Ziru, Xinying, Matthias, and Stefan Verger, who is our visitor right now, as well as all the previous lab members and our collaborators. And thanks a lot for your attention.
Thank you, Dr. Long, for an excellent presentation. At this point, I'd like to hand things over to my co-moderator, Abdul Kabir Abdul Malik, who will guide us through the next part of the session. Abdul Kabir, the floor is yours.
Yeah, thank you very much, Aslam, and really wonderful presentation by both speakers. Really exciting and insightful, I would say. So we have a list of questions already coming in as you were presenting your talks, and the first one goes to Professor Carl, and it's that during the hiring process, what are you looking for specifically? So.
Yeah, I think that's a great question. I think what you're looking for will depend on whether you're looking to appoint a PhD student or a postdoc. So typically, we will appoint a postdoc based on project requirements, so a particular skill set or particular expertise, and you need to specify that in the ad for the postdoc. Now, for a PhD student, it's a bit more general. Yeah, essentially looking for someone that intellectually capable, that has an interest in the project or in the topic of the research. I think that's very important. Yeah, I hope that answers the question.
Yeah, I think it does. Maybe Professor Yu Chen, you want to drop comment on that?
Yeah, I think actually indeed, I think a targeted search is very important, and then I think both yourself and the candidate should know actually what you're looking for. So I think maybe adding on to what Carl says, in our cases, we also actually try to see who are the candidates if they actually fits with the chemistry of the lab. So I think I actually really agree with what Carl said about trying to invite the candidates onto for a physical interview. And actually in Singapore, this could be kind of difficult because we're kind of far away from everyone in the rest of the world. But then I think when opportunity provides, actually we try to host in-person interviews. So not only can we get a better feeling with the candidates, but also can actually have our lab members to interact with the candidates on a one-on-one basis. And I think during this process, it's not only to see how actually the chemistry meets, but also it's a great way to actually pick everyone's mind to see how do they think the scientific quality, the way of communication as well. Is that actually a good addition to the lab? And yeah, I think it's quite important also to be consider on personal levels.
Yeah, thank you very much. Definitely, having a close fit with the projects are actually really important to consider when assessing individuals to join the lab. Thank you. Just going with you again for the next question, Professor Yuchen. What advice would you give for being scientifically productive when you're starting with a smaller group, for example, a PI and 1 or 2 students?
Yeah, so actually, this was exactly how we started with one PI, and then I think we first had one postdoc, who was Kartik Babu, and actually he was the last PhD student of the previous lab in the same space. So actually, Kartik was actually he knew the university, knew the department way better than I did, and I was very lucky to actually be able to hire him, who also had a very fitting background as well as the expertise. And also the chemistry also mix very well. So I think in this case, you definitely want someone who can help you in establishing the lab together. And then I think in this case, really being able to work in a team with a set, with one single set goal is going to be very important. If you can have someone who have experiences before, which sometimes can be very difficult, but then at least if you have people who are motivated in actually helping you to build a lab together, I think that it's very good way to go for. And I think these new lab members also need to be very brave to actually approach you for a new lab that has just been established. So I think mutual respect needs to definitely given back to your lab members, so then they also feel acknowledged, and then actually can build this whole thing together with you. Yeah, not sure if it's answering the question.
Definitely does, I suppose, because it's quite important. I think the challenge is majorly like when you're just starting out and you really want to decide the hiring process as well as being scientifically productive in the same context. So quickly for just to throw in this, Professor Carl, how do you handle a student or a postdoc who is underperforming in your lab?
Can you repeat the question again?
Yeah, so how do you handle a student or a postdoc who is underperforming in your lab?
Yeah, that's a really good question. I think having conversations is very important, trying to understand why the person may be underperforming. it's a two-way situation here. You not only need to understand the situation of the student or the postdoc, but you also need to understand yourself. So are you placing, undue expectations? Do you have such high expectations that you perceive them as underperforming when they're actually performing? So it's a two-way thing. So you need to evaluate yourself, but at the same time understand there could be multiple issues at play here. There could be personal circumstances that you're not aware of that may have impacted on their research progress. So it's really important to have those conversations, and if needed, refer them to professional help. I think that would be very useful.
Okay, great. Professor Yichun, do you want to add something to that?
Yeah, I think actually, indeed, I agree with Carl that underperformance is maybe something that is not perceived in the same way by different people. And I think in the end, as actually a manager in the lab, what is very important is actually the people themselves. So very often this may happen when, as we say, the goals are not aligned. So I think indeed talking to your lab members, align your goals. Actually, what is the final target? Do I want to excel in research? Do I want to actually move on to the next position, or what is actually the desire that is actually either in the way or actually diverging the attention? And I think this by actually aligning your things, and also depending on how you want to help maybe the lab member or actually help the whole culture, then this may actually help you to realize what is maybe missing out there to support your lab further.
Awesome. So a question just came in, which is like similar to the question of actually starting small and building on an exciting idea of a lab named by consensus. What is actually the sensible number of people to start to work on things like lab culture and lab coherence? When do you actually go from us to our lab?
Oh, that is a very good question and I guess, very difficult to answer. I think I don't have a clear answer to this. I think we're still kind of in between this space of us and our lab, and actually, I don't really know like what is the distinguishment between these two. But yeah, I think our lab kind of started from 1 person to 2 person to 4, and then now we are kind of like maybe 8 members plus internship students that also come in and leave quite frequently. I think starting from the beginning, we realized that actually there is a target goal that we are working towards, and then I think this is a moment that we're growing together. And again, if you will, thanks to COVID, maybe it kind of really helped us to actually glue together in a confined space. So, actually build up the group spirit very quickly. But later on, I think when new members come, they always bring new ideas and new energy. And I think the moment that we start to get used to, we're always welcoming new things. And I think this is where we start to become also very confident as a collection of people. And I think maybe this is where the whole lab consensus may have emerged. So I would say maybe at the beginning it felt more like a family, which I don't know really if it's a good thing or bad thing. But now I think we're now in a very, in a very nice position between being very personal as well as being very professional.
All right, thank you very much. So I'll just take one more
Can I
question
just
because
follow
of
on from Yichen?
Oh,
Sorry, Abdul. Yeah,
yeah, perfect.
I think Yichen gave a great answer, but I don't think there should be a separation between us and our lab. I think they both mean the same thing. Essentially, it's the belonging in the lab, and lab culture will evolve. When you have new members joining, and when you have new members from different cultures joining, the lab culture will evolve as well. But the underlying principle is really scientific integrity, and you cannot get away from that.
Yeah, that's perfect. Perfect. So we actually have to wrap up here. And for sure, if there are any more questions, we would see if it would be possible to kind of answer them at a later time and send it to the attendees. But for now, we'd like to really, really do appreciate both our speakers for excellent work done. It's really, we say thank you very much for taking your time to come here and share this useful and very insightful nuggets with us, and for the insightful answers as well. So now I will hand over to Jason to wrap up this whole session. And yeah, thank you very much.
Yes, yeah. Thank you, Abdul Kabir. Again, thank you to our speakers and to the rest of the moderator for today's webinar. You'll share so many great insight with us, and so we greatly appreciate it. As a reminder, this webinar is recorded, and you're able to watch the recording of it on our Plantae YouTube channel. And materials will be posted in the Plantae webinar blog once it's processed. With that said, I hope everyone have a great rest of your week, and we hope we'll see you on our next webinar. Bye, everyone.
Thank you. Bye-bye.